compounds of HIV and a target for therapeutic drug designs. The enzyme itself consists of 3 domains. First, the N-terminal domain that has a his2 cis2 motif that chelates zinc. Next, there is a core domain with the catalytic residue sequence of DDE motif, which is required for efficient enzymatic activity. Lastly, there is a C-terminal domain that has a SH3-like fold that non-specifically binds to the DNA. There are 5 characters, each playing one individual role in the skit you're about to see. Brian and Connor are both playing the part of Integrase dimer. These parts of the mechanisms will be portrayed as Andrew's friends. Andrew will be acting as the viral DNA, which is portrayed as himself. Kurtis will be one half the chromosome, which is portrayed as Destiney's boyfriend. And Destiney will be the other half of the chromosome, which is portrayed as Kurtis's girlfriend.
You guys, what do you say we hit the town tonight? Andrew, you're new in town. What do you say we show you around?
Hey, I don't really know about that. Why don't we just stay in and get a red box or something?
Nah, man, that's lame. You gotta learn to get out of your capsid sometime. I hear there's this awesome restaurant called The Nucleus. Head up sometime. You gotta get into the social scene.
Retroviruses are a class of viruses which replicate themselves through a unique process of reverse transcription. One example of a retrovirus is the perversive disease known as human immunodeficiency virus, or HIV. A reverse transcriptase codes the viral DNA to DNA. Complete viral replication would not be possible without integrase. HIV-1 integrase is a key part of viral replication process. This enzyme binds with reverse transcriptase and accepts the complementary viral DNA. The catalytic site of integrase incises the host cell's DNA and inserts the viral DNA. The integrated viral DNA is replicated during the cell cycle and subsequently passed down to the daughter cells. Integrase is a key component of virus propagation.
Hey girl, check this out. What do you think of these jeans?
Sorry, but my genome's full.
Oh man. Joe, I think there's more DNA over there. Let's go check that out.
Yo, Andrew, having fun? Bet you are. This place is great. Told you. Listen, I know this couple. I've known them for years. They're the top of the town. Everyone knows them. They run this place. So in order to kick this place into high gear, I need you to help me to break them up so we can make all the decisions around here. What do you say?
Man, I don't really know about that. It just doesn't seem right to always be breaking up couples.
Dude, Andrew, we've been doing this since the 80s. We're professionals. If we can do it with monkeys, we can do it with people.
Agreed.
All right, if you guys are as good as you say you are, I'll do it. I'm just a little nervous, so could I have some water first?
Yeah, sure, man.
Thanks.
This scene represents the 2 dimers of integrase transporting the viral DNA into the nucleus, which is pre-integration complex, preparing the viral DNA for an attack on the target DNA by first bridging the pre-prime ends, then cleaving the dinucleotide from each end, leaving CHON bare on each 3' end. Water acts as a nucleophile and cleaves the dinucleotide TG from 3' ends of the viral DNA. This is why Andrew needs a drink.
You loose? Well, aren't you the most important macromolecule around here?
Well, that's very flattering.
Hey man, no one's talking to you. Why don't you get out of here?
You know, maybe I will.
Go get a drink.
This scene represents the binding of integrase complex with the target DNA. This is the formation of the transient target capture complex. The integrase making the phosphodiester bonds of the DNA backbone is susceptible to nucleophilic attack from the 3' OH ends of the viral DNA, and the subsequent attack by the nucleophile at the same time as the opposite end of the DNA backbone is broken away. This is ushered in the formation of the strand transfer complex.
Destiney, this is our friend Andrew. I think you guys might have a lot in common. I think you should get to know each other.
Yeah, Andrew, why don't you take a seat?
Thanks. By the way, do you like jeans? I have some pretty novel ones.
Several bad puns later.
Man, that really hit the spot.
Yeah, thanks for dinner, Andrew. Do I have anything on my face?
Just a few primers.
Did I get them?
Oh, you missed them.
Oh, let me take out my mirror.
The purpose of Destiney looking into a mirror and noticing that she looks different is reference to the inversion of chirality that has come about by the SN2 reaction during transesterification. This scene represents the removal of dinucleotides from the unpaired 5' end of the viral DNA by cellular enzymes in there, which is 5' ends, binding to the 3' ends of the unpaired target DNA. This is represented by Kurtis, who's just returned from the bar.
Yo, man, I see Kurtis come in. Let's make like a halogen and get out of here. Hey!
Hey man, I'm sorry about my friends. They can be kind of subversive sometimes.
Well, I don't know what subversive means, but I accept your apology.
Why don't we take a selfie together then?
That sounds like a great idea.
The selfie represents the translocation that has occurred and shows Andrew between Destiney and Kurtis.